Welcome to another episode of the Nuclear Medicine and Molecular Medicine podcast. So we're going to do something a bit different today. I'm going to be interviewed because I'm one of the authors of this paper, and I'm actually going to be interviewed by one of the other authors, and that's Dr. Katie Davey from the Biomedical Engineering at the University of Melbourne. So Katie, take it away.
Excellent. Okay, so I guess to set the groundwork of this study, it would be good to introduce who we are and use in the context of what was the motivation for this study.
Sure. Well, what we are, we're the Melbourne Brain Centre Imaging Unit. We're part of the University of Melbourne, but we do lots and lots of Alzheimer's studies with tau and amyloid and all those kinds of traces for clinical trials or for basic research into Alzheimer's disease. And we were one of the first places to use in large numbers the MK tracer, the new tau MK tracer. And we also were one of the first places to use in large numbers the more traditional first generation tracers, the 1451s and so on, the AVID tracers. And I was talking to the physician in charge of that, Professor Chris Rowe, and I said, all these blobs of tau sticking up everywhere in the skull and everywhere else. It's a bit hard to figure out what's important and what isn't. He said, yes, totally. I don't know how we can figure out what's important. I said, well, how about this? Why don't we let some, computer algorithm try and figure that out? Perhaps if we compare it to amyloid number, we can see which bits of the tau are important and which aren't. And that's where this all started.
So just as a quick background then, to introduce machine learning and deep learning. So deep learning being a kind of machine learning where we train the computer to identify the important bits of the image. So we train the computer to extract the features of the input image, which is a tau image, and tell us which features are good at predicting the output, which is the amyloid, which in this case is a centiloid number. So using deep learning, we train the computer to tell us which of the input features are good at predicting the output. So that's the engineering part of this. And now maybe you can tell us what we did from the more clinical perspective.
Sure. Well, there's two things that happened which were very surprising, I guess, is that we got a whole range of subjects who had a whole range of disease, from healthies right through to people who had Alzheimer's disease. So the complete spectrum of people who had a whole range of amyloid numbers, over 100 of them. And then we also had those people that had had a tau image with the MK tracer quite close in time period to that. And then we put that tau image in, and we didn't give any prior assumptions. For decades now, we've had prior assumptions about what happens with tau. They followed the pathology model with these BRAC models to try and see how closely our amyloid PET imaging or tau PET imaging followed these BRAC models. But we ignored all of that. We just got an amyloid image. We've generated a centiloid number, and I won't go into that, but that's a generalized measure of the overall amount of amyloid. And we did that with a NAV tracer, and which is, probably the best amyloid tracer. And the other thing we did do was we knew that we'd be looking at little spots of very small areas of tau. We did our best resolution pictures. So we used better quality pictures than what are used in things like the ABLE trials and the ADNI trials. So we used high resolution PET scans with a high resolution kernel so that we had better quality pictures. And we knew from our previous studies and from previous stuff that by doing that, we could pick up very small amounts of tau that can't be seen if you blur and smooth an image to make it visually appealing or similar to what's used in things like ADNI and ABLE. And what we actually found was that you could very, very accurately, with a very straight, narrow line from a tau image, predict the amyloid.
Can you tell us what the potential limitations are with the models that use the assumptions that you're trying to avoid, such as the assumptions associated with the BRAC model?
Yes. So lots of our previous imaging has been based on optimizing it for visual interpretation. And people don't like to see noisy images. But noisy images actually capture what's really happening in the body. It's a noisy process. And so they actually lower the resolution by smoothing the images to make them more visually appealing. So we didn't do that. The other thing we didn't do was we didn't try to make our PET images. We didn't set up a whole lot of regions similar to what BRAC and so on have been done in the last 20 years previously. And a lot of the scanning with PET scans, particularly for tau, has been trying to make the PET scans fit into that pathology model. And pathology models aren't necessarily the gold standard, because you're not really measuring quantitatively material like you can in a PET scan. And the other thing is, we're not assuming those same regions in a PET scan. We got rid of all of those assumptions. We just wanted to see what the deep learning would reveal if we looked at the association between amyloid and PET.
So it's a purely data-driven approach with a very minimal set of assumptions to let the data drive the conclusions that we're drawing. Exactly.
And when we did that, we actually came up with things that we really hadn't seen before. And what we found was that you could actually look at amyloid elements in the tau image, could actually predict an amyloid number right back to -5 centiloids, which meant that really back to healthy people.
And one of the important aspects of our input was that it was scaled so that no matter what the amyloid burden was, the input images were all scaled to have the same range. And do you want to tell us about why we made that assumption?
Yes. We know that there's a lot of tau that accumulates at the end of the disease. So really high tau numbers, right? And we didn't want that swamping our approach right through disease. So what we did, we took each tau image and we scaled it to 1, right? And we looked at the distribution of tau in the image, right? Now, at low numbers which really have very low amounts of tau, we still looked at that the distribution of tau in those images and used that to try and predict the amyloid number. And much to our surprise, we found that even in images which were visually not very impressive, the machine learning was able to still use the data in that image to predict an amyloid number. Now, the assumptions that have been made previously is that we don't see tau until amyloid levels reach 15 or 25 centiloids. But actually, before that, we could still use the elements in that image to predict the amyloid number. So that means that there must be some changes happening earlier on in the images that can tell us about amyloid. That means that amyloid and tau are related right through the whole life cycle of Alzheimer's disease back to completely healthy people.
And importantly, we were able to use this same trained network for any centiloid value, although the network could implicitly be using different regions as the centiloid number changes, but it wasn't necessary to threshold the centiloid and do training for each cohort separately.
That's right. We could do it right across the whole range. And I think what was interesting is that we then went a bit further, right? We actually then decided to figure out what parts of the image were key.
Mm-hmm. So can you tell us about how we did the training of the network to make sure that we weren't overfitting or in some way using the training to help us with the validation? So we have to, I know that people can be quite critical of artificial intelligence or deep learning type networks, because if your validation set isn't completely separate, then you don't know if you've overfit to noise or other aspects of the data. So can you tell us about the approach we did for the training?
Great question, Katie. Yes, that is one of the main criticisms people have of deep learning. Firstly, that you do overfitting, and firstly, that it's not generalizable, right? So I'll start with the second bit first. It's not, people say it's not generalizable. Well, we didn't test that. We didn't want to look at that. We wanted to look at what the deep learning could tell us. So we did everything on the same machine. We did it all with the same details, so that you can't be criticizing to saying it's not generalizable, because we're really trying to understand what the deep learning model told us.
And by generalizable here, you're talking about across different scanners, right?
Different scanners, different modalities, different traces. We use the same tracer. We use the same scanner. We used all of the same settings. So we didn't try and make it generalizable. We just tried to understand what was going on in the biology. The other thing that we did do is we kept a sample of the same data set out and didn't use it for training or anything else. We just used it for validation, and we then went and put those tau images back in after we'd completed the model to see whether it would still predict an amyloid number, and it did.
So those images were not seen during training or testing. They had never been seen before.
They'd never been seen.
Naive.
What was more is we didn't look at just the best model that the deep learning model. We looked at all of them. Right, and all of the different models, actually, even the not so good models at predicting amyloid, all gave the same regions were important, the same clusters were important in predicting the amyloid number. So not only weren't we guilty of overfitting, but we also weren't guilty of selecting the best model to show the results.
And we had multiple models because we used a K-fold approach, right? So we had a K was 10 in our case, so we trained 10 different models and then tested to see how consistent they were in the regions identified as important.
Exactly. So I hope we've answered anybody's critics of the deep learning model, because really we weren't so much interested in seeing if we could predict one from the other, although that was important, but also what it would tell us about the underlying biology. And we found that there were certain regions that were important at different levels of amyloid. We found that at low levels of amyloid, some regions were important, and at higher levels of amyloid, other regions were important.
And how did we tell? Because obviously, deep learning network is a black box. Yes. So how did we then sort of reverse engineer to identify which regions the network was using?
Yeah, that's a great question, and I think that's the great answer, that we could use this in a variety of other biological models too. What we did, we hid from the network parts of the image, right? We said this is one cluster, this is another cluster. We hid different clusters from the model and said, now how well can you predict? And we found that some clusters were important early in the disease process. If you excluded all others, they could predict the outcome of amyloid just as well, or virtually just as well. And other clusters were important later in the disease process.
So this is called occlusion analysis or occlusion testing, right?
Correct, occlusion testing. So that meant that we could track the association of amyloid tau across the brain. So it went from the cingulate areas and the frontal areas into the medial temporal areas. And we found that even though you've seen lots of other papers that said, oh, the tau goes everywhere and everything else, really those very small areas were responsible for giving all of that information about how it related to amyloid. And we suspect that once the amyloid's gone out from the medial temporal areas, once the interaction between amyloid and tau stops at the medial temporal areas, tau then spreads out on its own without the influence of amyloid. So it tells us something about the biology of what's happening, the localization of the biology of the amyloid and tau. And that's something that hasn't never been seen before. People assumed that tau came along later, many decades after amyloid appeared. And it seems that they're tightly bound and they've got a tight relationship. And that is now borne out by the clinical findings of therapies for amyloid and tau, saying that really they're not separate items, they're part of the same process. It's just that we weren't really looking properly for those low levels of tau. We weren't doing our PET scans properly. We weren't using the power that the deep learning could give us to tell us what's happening in the biology.
Yeah, the prior assumptions were getting in the way of the data analysis, I think.
Yeah, and often a big culture goes along and it almost becomes a dogma, I think, sometimes. It's useful where a deep learning approach throws that dogma out the window and says, let's just see what the data tells us. And I think we've learned from that.
So do you want to discuss the comparison of our deep learning based with the standard GLM approach?
Sure. Instead of just looking at the uptake of tracer, which is what happens in the sort of usual model where we try to look at regions and look at the uptake and compare the uptake, that'll produce a weak association, but we found a much stronger association by using the deep learning. And we think is the deep learning might use several regions, right, to associate an uptake. One might act as a reference region compared to another, or the ratio between regions, or three-dimensional views across the whole volume of a cluster of regions. And the combination of those is what's predicting the outcome. Those are the kinds of things that might be happening in a deep learning model. When we just look at an individual voxel or a picture, we can't see in 3 dimensions. We can't look at multiple values. So that's the weakness of our traditional way of looking at things. We only look at one spot and look at the increase uptake. Now,
so the GLM does an individual voxel-based approach where each voxel is examined separately and independently. So we can't then account for any dependence between the voxels or the regions, which a deep learning approach can do.
The deep learning approach can do that. And just because an area is significant in deep learning doesn't necessarily mean there's a high uptake of trace.
Mm-hmm.
Right? And I think, and in fact, we showed that. We actually looked at different regions, said some of these have got high uptake and some of them have got low uptake. But it's the combination of those regions that tells us the story. So don't assume that just because we said a region's important, that it's necessarily high uptake. It just means that the combinations of those regions are important in predicting the outcome. So that's the subtlety you need to do when looking at this, the deep learning approach, and the power of it, because it means that the 3D networks, the association between parts of the brain and networks, like people have seen in MRI, applies also in PET perhaps, and that way that we can look at that network approach to brain to predicting the biology. And I think that's a really clever way to do it. And I think, we perhaps can use this in other ways too.
So before we get on to applying this technique to other clinical applications, do you want to tell us what the main implications are for Alzheimer's in your opinion?
Got rid of the old assumptions that amyloid and tau are actually happening and interacting at the same time. It's just at the very low levels of tau in the beginning, and that those regions aren't constant. They're changing across the brain. And that gives us a clue to the biology of the disease, and it gives us a clue to therapies. Are we going to attack the therapy where that tau and amyloid interacts at different times of the brain? Are we going to use imaging to actually figure out whether that therapy is working and how it's working? Not just by looking at uptake, but that interaction between tau and amyloid. If we can break down that interaction, perhaps we've got a better clue to a better therapy, and perhaps we can understand the origin of the disease process. Perhaps when we do other therapies, like with vaccines to prevent amyloid accumulation, we can see whether it's working by seeing whether it blocks that interaction with tau, for example. So there's lots of ways we can use this technique to see the effectiveness of a therapy. And I think that's some of the clues that we've had by not thinking in one dimension, not thinking about just one protein and how it accumulates, but how it relates to the whole disease process. And I think there's other ways we could use this as well.
So what do you feel like the most immediate extensions or next steps for this work?
I think the next step would be to expand it beyond just amyloid and tau PET images. We could do it, for example, with amyloid and tau images and relating to a biological marker like neurofilament light to see where we're looking at damages, or relating CSF or blood-based biomarkers to an image to see where they're interacting with the image for Alzheimer's disease. And so I think there's lots of ways we can do on that. And then we can also use it to look at some of the new therapies that are being done in Alzheimer's for both tau and amyloid, and seeing where they're interacting in the brain and when they're interacting in the brain, so that we can see early on whether a therapy is working properly by stopping the production or changes in tau. And that's important because we know that changing amyloid now does affect tau, but when and how, not just the uptake, but that early interaction. We don't want to have to wait 5 or 10 or 20 years in a therapy to figure out whether it's working. Using this deep learning approach, we can look at very subtle changes in tau to see whether a therapy is working, and by measuring it against some biological markers of those things as well.
Do you think it's necessary to confirm these results on a different scanner or with a different imaging modality?
We certainly could, and I think that is worthwhile. I think you could use a different scanner and a different tracer. But I guess it's going to be very difficult to generalize these things with deep learning. I think you don't want to use this as a way to replace humans. I mean, a lot of the deep learning models have done to replace, but we're not talking about replacing humans or replacing something else. What we're talking about is learning from the model itself and figuring out, understanding the biology. So I think that would be more important if we're going to try and generalize it. But if we're trying to actually understand the biology of a therapy, then we don't need to generalize it, right? We can, as long as you've got your samples and your data and your traces similar, then you can actually understand the interaction. But I think it'd be great if you do that. I think that's why it's important also to have good numbers on a particular scanner and a particular tracer before you go on. Deep learning does require significant numbers. So I think that's an important lesson we can learn from that.
And do you think this approach can be applied more generally?
Yes, I think that's one of the most exciting things. I see no reason why you couldn't, for example, in oncology, look at how it relates to blood or other biomarkers in the body. For example, changes in the immune status of checkpoint inhibitors, using checkpoint inhibitors to establish a therapy against an image. What elements in the image, right, of an FDG scan or a prostate marker scan or a neuroendocrine tumor scan, what elements in that image are most important at predicting a therapeutic outcome by measuring against a therapeutic outcome or by measuring it against a biological blood marker for immune changes. Or you could use it in cardiology, for example, trying to measure what elements of an image are good at predicting cholesterol numbers, or what elements are good of an F- image are good at predicting heart attack changes or heart perfusion changes. So we can look at the whole biology of a patient by combining not imaging and other biomarkers together to see what's important and how those interrelate and where they interrelate in the body and when they interrelate in the body. So when does cholesterol impact the changes in heart disease, for example? When does your PSA make changes in prostate cancer? All of those. When does Herceptin make changes in breast cancer? There's lots of things we can do with this approach, and I would encourage researchers out there to take this model and use it in whatever field you're in, to ways of combining imaging with non-imaging biomarkers or with other imaging biomarkers to understand the biology of disease you're trying to follow.
Okay, so where can people find out more about this work?
Okay, well, I'm going to put a link in the thing to our paper. But remember, we're not the only people doing deep learning, right? And deep learning's been used a lot to develop generalized clinical outcome tools, generalized clinical tools. I think what we've done, it's a little bit different, is we've looked at the trajectory and location on an image of where that interaction happens. Other people have looked at images too and used those images to predict things. But I guess our particular technique where we try and look at the trajectory of change and then we try to break that down into the location is a little bit different. And I think you can use that to figure out where those biomarkers are interacting and when they're interacting as well at the same time. And I think that's
the deep learning was just the first stage in this research in giving us a clue as to which regions were critical in the prediction of amyloid from tau. And then the research involved another whole phase where we then analysed each region and its impact on amyloid. And importantly, never at any point did we look at each voxels independently and treat them as a set of independent systems. So I think that's the thing that's critical about our research and maybe sets it apart, is that the deep learning was just the first stage, and then it was followed up by a full data analysis component that helped us then assess the impact of each region and the importance of each region.
Yes, that's what made us a bit different, and I think, and importantly different. I think that we should learn from that to actually develop ways we can apply this in other spaces, and that is not just do the deep learning, because I think deep learning is used a lot, generalised it'll be this way, but understand how the deep learning came about, and how we can use that to identify when and where that deep learning had its effect. We know deep learning is very good at making predictions. What we're really trying to do is figure out how it made those predictions.
Yeah, absolutely. And I think that's critical, because as everybody knows, deep learning is a black box, and it's what you do with the network after it's trained that is really crucial.
Yes. Excellent.
So will you include a link to the paper and to the research site so that people interested in following this workup will be able to find it, and the research team?
Absolutely. I'll include a link in the podcast for that. It was published in Nature Scientific Reports, and we'll include a link to that, and we'd be happy if you get in contact with us to discuss more. Perhaps there's ways we can combine together and look at other ways of researching this, and I'm sure you'll find that of great interest. And there's lots of researchers out there in a whole variety of fields who could benefit from this kind of approach, I think.
We should also mention that the work was largely completed by our PhD student, Gihan. I'm not sure if I could pronounce his surname with any confidence, but Gihan was the PhD student who actually completed the majority of this work. So.
Yes, we've got a great PhD program here at the University of Melbourne, which combines engineering with medicine, which is really good, and PET and so on. And we've got a fairly unique setup here with a university-based PET scanner for doing this kind of research. And I think it yields different results than what you might see in some other, more medical approaches, I think.
Yeah, absolutely. Well, thank you, Rob, for participating in my podcast.
That's appreciated.
Thanks very much.
And have a excellent season, everybody out there, and have a good break over the holidays.
Merry Christmas.
Merry Christmas.